Okay, so let's do a quick review of the action potential and what causes the action potential, which would be a graded potential. So here's a nice figure of what an action potential looks like. Let's kind of look at the couple things. First, let's look at the axes. So we have the x-axis on the bottom, that's time, and notice the window's only about 5 milliseconds long, so the action potential is a very quick event. Look at the other axis, you have membrane potential, which is also known as membrane voltage, and that's given in millivolts. If you look at the action potential, well, it's not exactly shown here. The minimum number shown on the graph is minus 90 millivolts, and the maximum up here would be about positive 60 millivolts. The reason why we constrain the graph with those numbers is we're just looking at the effect of sodium and potassium in terms of the action potential. So if we're looking at sodium, sodium, if you remember, has an equilibrium potential of positive 60 millivolts. That means that when the cell is permeable to only one ion, in this case sodium, the voltage would be positive 60. On the other side, down at the bottom, if the cell is only permeable to potassium, the equilibrium potential for potassium is minus 90. So if the cell were only permeable to potassium, it would be at a value of about minus 90. I'll just kind of illustrate that a little bit further. What would be the membrane voltage if the cell was half permeable to potassium and half permeable to sodium? Well, the easiest way to figure that out is to look halfway between minus 90 and positive 60 up here, and that'd be right here about minus 15. So that'd be the halfway point if the cell were equally permeable to potassium and sodium. So looking at number 1 on our graph, that indicates the resting membrane potential. That shows minus 90 millivolts, which means the cell is more permeable to potassium at rest than to sodium. It's not completely permeable to potassium, otherwise it would be at a minus 90 millivolts. So in reality, about 97% of the channels that are open in the cell membrane are open for potassium. Only about 3% are open for sodium. So we're just moving along from 1 to 2. That's our resting membrane potential. Now, if you look on this from 2 to 3, and it's indicated here on the graph, that is your graded potential. So we mentioned graded potentials before. We have two basic types, the inhibitory or IPSP signals and the excitatory, the EPSP signals. Given that this one takes us more towards threshold, so we're going in the direction towards threshold, this would be an excitatory stimulus, meaning we're more likely to have an action potential in this case. So what are the 4 ways we can make an excitatory stimulus here? The 4 ways would be we can open a sodium channel. More sodium ions would come in. Since they're cations, that would make the cell more positive. So that could be happening from 2 to 3. We could do a similar thing with calcium. Since calcium has a high concentration outside, a low concentration inside, we open a channel for calcium. Calcium diffuses in. It's a divalent cation. Remember, the charge on it is +2. So calcium will come in and make the membrane voltage become more positive. Or we could alternatively close a potassium channel. Remember, potassium has a higher concentration inside the cell than outside. Since potassium has that higher concentration, if you open a channel, it would diffuse out and the cell would become more negative. So if we close that channel, by relationship, then the cell would become more positive. Likewise, if you close a chloride channel, chloride, which is an anion and highest concentration outside, so chloride would be diffusing in. If we close that channel, we'd have less negative ions in, which would give us a positive deflection in terms of the membrane voltage. So those are the 4 ways we could generate this graded potential, or a combination of them as they're additive and they can summate. Open a sodium or calcium channel or close a potassium or chloride channel would make us go from 2 to 3. As we mentioned before as well, The magic number we're trying to hit is that dotted line that says threshold. That's at minus 55 millivolts. At minus 55 millivolts, that's when we open the first double-gated sodium channel in the axon hillock. Functionally call that the trigger zone, and that generates the action potential. Once I open that first double-gated sodium channel, sodium's going to come in, and as sodium comes in the axon, it makes a positive deflection. Once I reach threshold, then what I'm going to do is I'm going to open all the successive sodium channels, those double-gated sodium channels in the axon, which is going to propagate the action potential from one end to the other. So from 3 to 5, which is actually shown here at number 4, right? So here's our number 4. That's actually the action potential. That's also called depolarization. The depolarization is primarily a result of an increase in sodium conductance. There's more sodium coming to the cell, making it more positive, so we get that positive deflection. The peak of the action potential is about positive 30, as you can easily see there. That action potential peak is pretty much the same in everyone under the vast majority of conditions. So the action potential is said to be all or none in an individual axon. It either reaches threshold and fires, and if it fires the same way, or it doesn't reach threshold and nothing happens. So 5 would be the peak of the action potential. Going back down the other side, from 5 to 7, so actually shown in number 6, would be the repolarization. Right, so we're going from this peak in number 5 and going down to number 7, so that's repolarization. Repolarization is primarily due to a consequence of sodium channels closing and potassium channels opening now, which brings our membrane voltage back down. Associated also with this initial phase of repolarization, from about 5 to this peak at plus 30, down to about minus 30 right here. So this range right here, is the absolute refractory period. The absolute refractory period has to do with the configuration of that double-gated sodium channel, where it's in basically a locked configuration, and you cannot get it to open and send another action potential for a given amount of time. So that's called the absolute refractory period. Going from that, just kind of finish the concept, from minus 30 all the way through back to where we get to resting membrane potential again. That would be the relative refractory period. Compared to the absolute refractory period, where again in the absolute you absolutely can't send an action potential. In the relative refractory period, if you give a bigger than normal stimulus, you may be able to generate an action potential. So the absolute refractory period is associated with repolarization only. Repolarization going on this side. And the relative refractory period is associated with repolarization and then some of the hyperpolarization as well. So those are the differences. Once we get to number 7, we've gone past our resting membrane potential and actually farther away from it or more negative. This is due because the amount of potassium channels open at rest compared to the sodium channels is greater during this portion of the action potential, and that's why it has a more negative voltage. Remember, the closer we are to 100% of the potassium channels open, the closer we will be to minus 90. So the hyperpolarization is due primarily to the increased percentage of potassium channels open compared to sodium channels during the recovery of the action potential. So from 7 all the way pretty much close to 9, this area is called the hyperpolarization, and we begin to go back towards normal. Once we get back to 9, again, we're at the resting membrane potential. So just a nice little table to kind of review the differences between action potentials and graded potentials. So let's start with the individual action potentials and kind of look at some of the attributes of it. In an individual action potential, the potential is all or none. If we compare that to graded, the potential and the graded potential is graded, which makes sense because that's what I call a graded potential. The amplitude for an action potential remains constant. It starts at a rest of -70 and reaches a peak of +30 if it fires, while the graded potential decays with time and distance. The farther we are physically from that potential, or the farther we are in terms of timing, that potential is going to be smaller. On location, the action potential is always on the axon, right? So always on the axon, and the graded potential could be on the cell body, dendrites, or axon. It really could be on any portion of the neuron. Coding refers to how do we signal the intensity of a stimulus. How do you tell a big stimulus from a small stimulus? And for action potentials, since they're always going to be the same size, they're going to be the same amplitude, we code for that by frequency. So generally speaking, the higher number of action potentials in a given amount of time, the higher frequency of them, that's going to result in a information stream coming to the brain, which is going to allow it to interpret what that frequency means in terms of the intensity of a stimulus. For graded potential, the amplitude, the size of it, indicates the actual strength of the stimulus. So a large graded potential would have a large amplitude, and a small graded potential would have a small amplitude. Channels with the action potential always uses double-gated sodium channels. They're the only set of double-gated channels in the human body. While single-gated channels cause graded potential, they could be single-gated channels for sodium, potassium, calcium, or chloride. The refractory period is a consequence of double-gated channels. Since the individual action potential has double-gated channels, it also has a refractory period. In terms of graded potential, since they're single-gated, not double-gated channel, and they can open or close whenever they're stimulated, it has no refractory period. For summation, in individual action potentials, they cannot summate because of the refractory period. They can't add together. For graded potential, you can do both temporal or spatial summation within the graded potential, and you can move those different graded potentials together and get an overall bigger graded potential that may reach threshold if you get it big enough. In addition, another thing to remember is that the excitatory graded potentials, the EPSPs, make it more likely to send an action potential, while the IPSPs are inhibitory. So, you could have, two EPSPs combined with two IPSPs and essentially have no net change in the membrane voltage because they've summated together. So the summation is not always going to be a positive summation with the excitatory stimulus. It could also be the negative inhibitory stimuli. In terms of the polarization, the action potential is always depolarizing. It always goes to +30, and it always goes in the same direction. For the graded potential, it could be depolarizing, taking you towards threshold. That would be an excitatory stimulus. Or hyperpolarizing, which is a stimulus that takes you away from threshold, and it makes it less likely to signal an action potential. So it's kind of a nice little comparison and summary between the individual potentials, action potentials, and the graded potentials themselves. This is just kind of an example of the frequency of action potential determines the intensity of a stimulus. What it tries to show is that a low frequency of action potentials, there's a small amount of neurotransmitter released. If we increase that stimuli, we'll have more stimuli above threshold, which means we'll have a higher frequency of action potentials in terms of that. So that's, again, an example of that rate coding we mentioned in the previous slide. One of the things that we should talk about a little bit is the conduction velocity, and sort of to get into that, let's talk a little bit about the size of the different axons. So axon diameter, we'll see in a minute, plays a very specific role in terms of how fast an action potential moves down an axon. When scientists were looking at classifying axons in terms of their size and speed of conduction, there were two groups of people that were actually doing it. One set were physiologists that study the motor side, so the motor system in those axons, while at the same time, a different group of physiologists that really, surprisingly didn't talk with or communicate with each other much. We're studying the sensory side. And so basically, if you divided the axons up into categories, we can say large, medium, and small. The motor people said, okay, we'll call the A the largest, and the B the middle ones, and the C the smallest. In fact, they even subdivided the different motor fibers, the type A. They have A alpha, A beta, A delta, and things like that. So they have subdivisions within that. Sensory people had a similar idea, but they actually, instead of going with a 3-tiered system, they went with a 4-tiered system. So the largest, again, was called type 1, and the medium ones were type 2 and 3, and the smallest ones were called type 4. In addition, by convention, the small fibers that are in the last category, the C fibers for the motor, the type 4 for the sensory, which are basically the same size fibers, are all by definition unmyelinated. So sensory smallest unmyelinated fibers would be type 4, and that would be the IV, right? And the motor ones, the smallest would be the type C fibers. So if you look at just the conduction velocity, how fast does the action potential travel from one place to another? In the largest fibers that would be myelinated, the conduction speed moves as fast as 125 meters per second. So 125 meters per second. While the small fibers, they move significantly slower, like maybe 250 times slower, where their speed is sometimes only a half a meter per second. So they're the slowest fibers, and that's how come sometimes like a withdrawal reflex. If you touch something hot, you'll move your hand away before you actually interpret the pain, because the reflex action of moving the hand is mediated by large myelinated axons. So that conduction velocity is fairly quick, while in the small axons, they mediate the pain sensation. I guess technically it's perception, but they mediate pain. And so those are much slower, and oftentimes before you actually realize it hurts, you've moved your hand away to reduce any tissue damage. So large myelinated axons would be the fastest, and small unmyelinated axons would be the slowest. And those would be, again, type C or type 4. And as an example, it's the type 4 sensory fibers that are the targets for anesthetics that we try to deaden a nerve, and actually something like lidocaine would block sodium channels and prevent the transmission of an action potential in sensory neurons, where we would just basically block the sensation of pain. So a couple other things about the speed we can talk about. It's not just the fiber size, which we're really talking about is in terms of the diameter, but also the amount of myelin, so the insulation, and also the temperature plays a role. Although in humans that tends to be minor, because we tend to keep fairly similar temperatures. So kind of to look at it in a basic way, then the conduction velocity is determined by a number of different factors. Number one is fiber size, and the larger the diameter fibers, the faster the action potential moves. So here's a cross-section of what's called a squid giant axon. And so squids have this big, huge axon running through it, and then all these little other white circles on the side, those are smaller axons. You can see there's very, very, very wide variety of axon diameters. So if you just looked at this and measured all of the axons and how fast they moved, by far the giant axon is about 0.8 millimeters across, which is fairly significant in terms of that. So you can actually study that, and that was really the first neuron that we actually studied in any extensive research on, because it was the most accessible. So the larger diameter, so this big one right here would be the fastest, and then maybe one like down here, which is pretty big, right, towards the bottom. That would be not nearly as fast as the big one, but faster than like these little tiny circles you see here. So that plays a role. Second idea is the amount of insulation, or myelin. And myelin makes it go faster for a very specific reason, and myelin serves as an insulator. And so what happens is only the nodes. So remember, these are the nodes of RON-VA, where we see these double-gated sodium channels. And during the action potential, we have to open these up so sodium come in and make the cell positive. Well, what we get a chance to do then is we get to skip the next set of on the axon because of the myelination and go to the next node. And that myelin then prevents what's called current leak. So if you look at the myelin on successive Schwann cells or within an oligodendrocyte in the central nervous system, you'll see that there are periods where we have myelin, and then in between the myelin we have those nodes. And so what ends up happening is, since only the nodes exchange the ions, the action potential is triggered, up here from the trigger zone, right next to the cell body, and then it gets to jump from node to node. And that jumping from node to node is called saltatory conduction, and that makes things go much faster. And not a bad way to kind of think about that in a practical sense is it's like using the tab button on your computer instead of the space bar. So the space bar would take you one space, the tab would take you many. And so that's why it goes significantly faster. The third reason, which again has fairly small impact on humans, is temperature. Since the action potential is created by diffusion of sodium primarily into the cell, then the higher the temperature, the faster the sodium is going to move because it's got more kinetic energy, which means the action potential would go faster. So temperature plays a huge role in especially, what we call cold-blooded organisms. And in warm-blooded organisms, they don't play as big a role because the body temperature is maintained fairly similarly. But so if you want to increase the conduction velocity within an axon, you need a large insulated fiber with a high temperature, would give you the fastest conduction rate. We're going to look a little bit at the ANS and not too much detail, and see how that's going to influence some of the things that we've done. We've seen some of these things in lab, and we'll actually see them again when we talk about the heart and blood pressure, and even a little bit about how the kidney works. So as a quick reminder, the ANS, like everything else in the nervous system, is made primarily of two distinct branches, the sympathetic nervous system and the parasympathetic. The sympathetic is most dominant at exercise. It helps you to make energy and capture that energy and bring in oxygen and increase blood pressure and flow and things like that. So it's often called the fight or flight system because it gets your body ready for action. The parasympathetic tends to be more dominant at rest, and at more dominant at rest, the parasympathetic plays a significant role. Because of that, it's sometimes called the rest and digest system, or occasionally the feed or breed system, because it also helps reproduction. So those are the two basic systems within the ANS. Couple things then that you can kind of look at as well. They tend to work antagonistically, and so looking at the seesaw, as the parasympathetic goes up, right, the sympathetic would go down, or if the sympathetic went up, the parasympathetic would go down. So there aren't any normal instances in the body where they both go up or they both go down, so they're said to be antagonistic, which certainly fits one of the Cannon's postulates, right, in terms of homeostasis. The other thing is, because we'll see in a second, they are not always as antagonistic as you would think, because in many cases, some of our organs or areas that are innervated by the sympathetic system are not innervated by the parasympathetic system. So we have what's called tonic control. So tonic control, again, means that we have a constant level of activity going to a various organ or area of the body. So those are the first two postulates in terms of One of the exceptions to the antagonistic rule are the salivary glands, and we talked briefly before about Pavlov and feed-forward stimulation in terms of ringing a bell, feeding a dog, and having them salivate, and eventually having them learn to be able to salivate without the food and just the bell in terms of a certain form of conditioning. So the salivary response has both sympathetic and parasympathetic, and it's an antagonistic response generally, where the parasympathetic would increase salivation and the sympathetic would decrease it. But certain times when you're exercising, in order to basically lubricate your mouth and make it easier to get air in to your lungs, sometimes you release more saliva in that case. So that's due to a sympathetic stimulation. And so it likely has to do with neurotransmitter changes and things like that. But the bottom line is in certain cases, the salivary glands can both increase their output through the parasympathetic stimulation or sympathetic stimulation. So that's one of the few exceptions to the antagonistic rule for the ANS. Primarily, one of the big things we see with the ANS is this idea of what we call dual innervation. Normally, both branches of the autonomic nervous system go to an area and innervate it, and that's called dual innervation. There are a number of exceptions to that, though, and let's start with the easy one, even though it's on the bottom. And so we're going to talk about things that are called monoinnervation, where a single branch of the autonomic nervous system go to a target area. The only exception that is cited for the parasympathetic only, so this means no sympathetic, is the lacrimal gland. And it's still a little bit controversial, but it's apparent that even though sympathetic fibers go to the lacrimal gland, they actually pass through it, but don't actually terminate in it. So the sympathetic tends to have no effect on the lacrimal gland. So that's an example of parasympathetic only, where it's got monoinnervation. The sympathetic only monoinnervation is much more extensive, though. So here are things that are sympathetic only without parasympathetic stimulation. We've already seen in lab the sweat glands, and they're sympathetic only. Your adrenal glands and your kidneys are sympathetic only. The ventricles of your heart tend to be not exclusively, but very close to sympathetic only. Your adipose tissue, which stores your fat, is sympathetic only. Your blood vessels are sympathetic only. There are a number of researchers that have spent many years looking for parasympathetic fibers in blood vessels and never find them. And your erector pili muscles, the ones that cause goosebumps, are also sympathetic only. One of the things that makes the ANS a little bit different in terms of how it behaves is that instead of having a synaptic end bulb like we see on a motor neuron, it typically has multiple synapses we call varicosities. And so varicosities are sort of like beads on the string. So if you look at this autonomic neuron coming to an area, and you can see all these different beads, look like beads on a string. So these little, what they call varicosities, these bulging areas, have neurotransmitter in them. And now they can, each one individually release neurotransmitter. And so there's much more than a one-to-one innervation for that ANS, where in the somatic nervous system it's one-to-one, and if you lost a fiber, you would lose the nerve fiber. You would lose the ability to contract that muscle, where there's a lot of crossover because of these multiple varicosities in areas for the autonomic nervous system. One of the other things we probably should hit a little bit is the neurotransmitters. There are two neurotransmitters that predominate in the peripheral nervous system, the ones that are by far most common. Those would be acetylcholine, and so that's what we call ACH. If you bind to an acetylcholine receptor, that receptor would be called a cholinergic receptor, so that comes from the word choline. And there's two basic types, what we call nicotinic and muscarinic. And we'll talk about what that really means in a minute. In terms of the norepinephrine, which we often just abbreviate NE, those receptors are called adrenergic receptors. And it's sometimes hard to remember, but since norepinephrine has a scientific name in Europe, and a common name in the US called adrenaline, and its close, noradrenaline, and its close relative, epinephrine, is known as adrenaline, then the adrenergic comes from that. So if you just think adrenaline, then adrenergic doesn't come too far from that. And there are 4 subtypes of adrenergic receptors that are responsible for what are called the alpha 1, alpha 2, and the beta 1 and beta 2. So the acetylcholine receptors, and acetylcholine uses neurotransmitters, the most ubiquitous neurotransmitter in the human body. Ubiquitous means it's found everywhere. So obviously it is an agonist, right, for the cholinergic receptors because it excites them. Interestingly, there are also other agonists for cholinergic receptors. Besides responding to acetylcholine, the nicotinic receptors, not surprisingly, also respond to nicotine. The nicotinic receptors are found in skeletal muscles, where they're exclusively excitatory and lead to muscle contraction. They're also found in autonomic ganglia for both sympathetic and parasympathetic systems. So much of the autonomic nervous system, one fiber exits the central nervous system, and then synapses on another fiber, which then goes to the end organ. And so in the autonomic system, there's usually a 2-fiber system. There are exceptions, but in that first synapse, the acetylcholine is released into the post-ganglionic fiber. The muscarinic receptors are named muscarinic receptors because they respond to acetylcholine again, because they're subtype of cholinergic receptor, but they also respond to a chemical called muscarine. Muscarine is a alkaloid substance that you find in what you would call magic mushrooms on the street, and they have an effect on the muscarinic receptors. What's interesting is muscarine does not stimulate nicotinic receptors, and nicotine does not stimulate muscarinic receptors, but acetylcholine stimulates both. That's why they're under the heading of cholinergic receptors. Muscarinic receptors are found primarily in the end organs of the parasympathetic division, like the heart or the intestines, and we'll kind of come back to that idea in a second. The adrenergic receptors are a little more difficult to understand for a number of reasons, but they deal with a variety of receptors. And so just to kind of, before we go on and look at them, I want to point out a couple things. So if you look here, remember this is where we release the neurotransmitter here, right? So this is one of the varicosities we see. Here's my synaptic cleft, right? So the neurotransmitter is stored in the presynaptic fiber, right? On the postsynaptic membrane, we'll see there are a number of receptors. We excite those receptors, and then we get a response. What's interesting about those receptors is we have adrenergic receptors. We have the alpha 1, the beta 1, and the beta 2. Conspicuously absent from this list is the alpha 2 receptor. The alpha 2 receptor is actually located on the presynaptic membrane. So if you look, it's up here and not across the synapse. That's fairly strange, and it's one of the first receptors we're going to look at in this class, and probably the only one, in, except offhandedly potentially, was where the alpha 2 is. So we'll kind of look at what the effect alpha 2 is in a second. But in order to understand it, you have to remember it's on the presynaptic membrane. Okay. Now, these are all receptors that are proteins, and so they're subjected to those 3 things we've talked about protein receptors before: specificity, competition, and saturation. So let's start with the alpha 2 because it's kind of the different one, and that's located on the presynaptic membrane, so right here. Right? And its job is to inhibit excessive norepinephrine release. So it's what we call a feedback inhibitor. And how it works, it has a low affinity for norepinephrine normally. So when norepinephrine gets released in normal amounts, it's not enough to be interacted with the alpha 2 receptor, and nothing happens. But if we have a large amount of stimulation and get a lot of norepinephrine in the synaptic cleft, And once these receptors on the postsynaptic membrane become saturated, it doesn't do any good to release more norepinephrine because it's there, but there's no place to bind. So it's basically a waste because, as we talked about before, that norepinephrine is either going to diffuse into a blood vessel or end up being enzymatically destroyed, where now it's inactive, we'd have to rebuild it. So to try to prevent those situations, when we have excessive norepinephrine, now we can make up for the low affinity of the alpha-2 for norepinephrine by increasing the concentration. And when norepinephrine binds the alpha-2, it usually means it's in a really high concentration. And the job of the alpha-2 is to have a feedback inhibition to making and releasing norepinephrine. So it helps to limit the amount of norepinephrine we release at that time. The other two receptors, three receptors that we see on the postsynaptic membrane are receptors that you would normally understand. So each of these are located in different places, although they could be literally located right next to each other. So as an example, in systemic blood circulation, so like in your arm, the alpha-1 receptors are literally located right next to the beta-2 in some cases. So the alpha-1 receptors, the only place I want us to remember where they're located are in systemic blood vessels. And when they get excited, they cause the smooth muscle to contract, right, which leads to vasoconstriction. So the neurotransmitter norepinephrine binding to an alpha-1 receptor causes vasoconstriction. For the beta-2 receptor, we're going to worry about where they are in terms of cardiac muscle. There's also some in your kidney as well. And what they do is they have effects that help to increase the blood pressure, basically. And so we have these nice scientific names for them, although from a normal standpoint, what happens is we excite a beta-1 receptor in cardiac muscle. It increases the heart rate. It increases the strength of contraction, and it increases the conduction velocity, the movement of the action potential through the heart. But we have to have some fancy scientific name to indicate that. So positive means increase, and chronotropy, chronos means time. So positive chronotropy means that we're going to increase heart rate. In terms of the second one, positive inotropy, ino refers to strength. So again, positive is increase, ino is strength. So that increases the strength of contraction. Positive dromotropy, positive means increase again. Dromo refers to conduction velocity. So that means we increase the conduction velocity through the heart. So beta-1 receptors located in the heart have a positive chronotropic, inotropic, and dromotropic effect, which increase heart rate, heart rate, strength contraction, and conduction velocity. The beta-2 receptors, we see those on systemic blood vessels, coronary blood vessels, and in your bronchioles. What it does is it actually causes relaxation of smooth muscle. And in those cases, if you bind to a beta-2, you get vasodilation, or in the lungs, you get bronchodilation. So it's interesting that we have them in the coronary blood vessels, because if our heart needs more oxygen, we already extract a significant amount of oxygen from our heart in terms of our blood vessels, so we have to deliver more blood. And the way you can get more blood to the heart is you have to dilate the blood vessels. So when you have higher levels of epinephrine during greater times of stress or exercise, when you need more oxygen, that'll help open up the blood vessels in your heart and allow greater blood flow and increase the amount of oxygen we can extract from the heart. One kind of last idea for the sympathetic system. I mentioned that earlier, that there were typically a two-fiber system for both the sympathetic and parasympathetic system. And there's one kind of major exception that we're going to kind of look at, and that's in the adrenal glands. Remember the adrenal gland, sometimes known as the suprarenal gland now. It sits on top of the kidney, and during times of stress, then we stimulate the sympathetic neurons that go to directly to the adrenal gland, and that causes these chromaffin cells, so see them right here, chromaffin cells, to release epinephrine. That's interesting because epinephrine will also bind to those alpha, beta receptors we talked about, so alpha 1, beta 1, alpha 2, beta 2 receptors, and help support the action that norepinephrine's doing. So it's kind of an interesting thing. So it's the chromaffin cells in the adrenal glands that release that. One final note, if we're looking at muscarinic receptors in the heart, which are also there, and we stimulated those, so if we had an agonist, remember agonist causes the action, those would have a negative chronotropic, inotropic, and dromotropic effect on the heart. So it would be antagonistic to what the beta-1 receptors do. So that's it for what we need to know for this upcoming lecture on the nervous system.